Hello everyone, this is Dr. Maha Abdurrahman. Today I'll take you through the secret power of genes, the DNA, RNA, and how they're integrated into protein synthesis. That's an important and challenging part of the A-level biology for all school boards. Very interesting session. Stay up with me. So the DNA, guys, is a short term for deoxyribonucleic acid. So DNA is deoxyribonucleic acid. And the DNA stores all the genetic information. What do I mean, guys, by genetic information? Is the instructions that makes you look the way you are. All right? This is really, I would call it a magic secret part of life. All right, this genetic material, guys, surprisingly is found inside the nucleus of every single cell. Okay, so imagine, guys, you have the same copy of the genetic information in every single cell. Why this genetic code or this genetic information is really important, and it's actually very unique and very specific for each one of us. Now, this code or these instructions code for a particular protein. Why proteins are really important parts here? Because this protein, guys, could be hormones. These proteins could be enzymes that controls all the reactions happening inside your body. These proteins could be antibodies that could help you to fight diseases. I'm gonna give a quick example here just for you to link what I'm saying. So for example, guys, those who have the diabetes, okay, those who have diabetes, their cells cannot make the insulin hormone. Why their cells cannot synthesize insulin hormone? Because the DNA inside their nucleus, their nucleus lack the instructions for the insulin. So they lack the insulin gene. So the insulin gene is not in the proper code, right? It's not there. So their cells cannot make insulin. That's why they have diabetes. So that's an example of how important the genes are. So basically, guys, your cells are able to make a growth hormone, for example, because you have the gene that is required for making or giving the instruction for making growth hormone. I hope this part was clear. So a question that would show up here, guys, which is how the instructions inside the nucleus of every single cell, these instructions are inside the nucleus, but the protein synthesis happens inside the ribosomes. So there should be a messenger that carries the code from the DNA in the nucleus to the ribosomes in the protein. This messenger, guys, so this messenger is called the RNA. Let me show you here, guys, another diagram would make it much more clear. So look at this. That's the DNA here, safe and sound inside the nucleus. This DNA has the instructions, has the code for all the proteins you require to live. Proteins are made and synthesized inside or on the ribosomes. So this is the place where proteins are made. Now, the messenger RNA, which acts here as a messenger, that carries the message from the DNA up to the ribosomes, so the ribosomes can read this message. So this message here, guys, I'm gonna call it the gene. It has a specific code. Okay, this specific code has the instructions for making up a particular protein. This protein could be insulin, could be growth hormone, could be any enzyme. Now I need this messenger to carry to carry this particular code from inside the nucleus to the ribosomes. Now, a question, why can't the DNA all leave the nucleus up to the ribosomes? Because the DNA is too large. The DNA is too large to leave the nucleus. However, the messenger RNA, which is just a copied part of the DNA, okay? So this copy is a small single strand that can leave the nucleus to the ribosomes where protein synthesis can start. I always like to give an example that maybe would make it clear for students. Think of the DNA that has all the genetic instructions. Think of the DNA as a book of recipes. This is a very large book of recipes that has the recipes for everything. This book, guys, is kept in the library. But where we cook is in the kitchen, right? You cannot carry on this book from the library to the kitchen because it's a very large book and it's not allowed to leave the library. So what shall I do? I can simply copy the recipe I need. Let's say I want to cook pasta, so I'm gonna go to the page of the cooking pasta, so that's the pasta recipe, and then I'm gonna write it down. Write it down. Writing down, guys, is called transcription. Okay, let me give it a little more space. So copying or writing down, guys, is called transcription. So in the process of transcription, you're copying down the part of the gene that you need, just like how you have copied the pasta recipe out of your big book of recipes, okay? And then you can take this recipe into the kitchen where you can start to cook your pasta based on the instructions written in this recipe. So the process of translating what's in this recipe into actual pasta, into actual food, is called translation. So transcription first, copying the gene you need into this copy guide is called mRNA, messenger RNA, and then this messenger RNA is used in the translation process to make up your protein. This, of course, happens inside our kitchen, i.e. the ribosomes, if I'm talking about the cell and the DNA and the protein synthesis. I hope it's clear that way. This it's really important for you to really absorb what I have said, because that's the core of protein synthesis. If you don't get this, you will struggle in mutation, in molecular biology, in many other parts, in the AS level, in the A2, and in the university. So because this part is the most important part today, so I'm gonna, echo myself reading this part again to consolidate all what I have said so far. So the DNA molecule is found inside the nucleus of the cell, but the organelle that makes proteins, which is the ribosomes, is found in the cytoplasm, as you see here. DNA is too large to move out of the nucleus, so a section is being copied into the messenger RNA, remember? Okay, so this process is called transcription, and then the mRNA is small, so it can leave through the nuclear pore. See, guys, these openings are called nuclear pore. So this mRNA, messenger RNA, can leave the nucleus and joins with the ribosome on the cytoplasm, where it can start to make up the protein. This process is called translation. it doesn't sound as easy as it said, because there are more details into the transcription and the translation that I need a session for each to explain it and get into the bottom of things. But if you understand the idea today, that's a big, big gain, because then you will be able to follow up with me when I do the session for transcription and translation. Now, moving on, we will explain the structure of the DNA and compare it to the structure of the RNA before we are able to talk about the transcription and the translation in the next session. So, DNA and RNA both are polymers. What do I mean, guys, by polymer? We already know from the chemistry, polymer is a long chain made up of monomers. Okay? So just like necklace, this necklace is made up of beads. So if I'm gonna call that necklace the polymer, then one single bead would be the monomer. And then they're joining together to form the big polymer. So the monomer here in the structure of the DNA and the RNA, guys, is called mononucleotide. Mononucleotide. So this mononucleotide is my monomer here. Okay? One mononucleotide, guys, is made up of 3 basic structures. Phosphate group, chemistry. Pentose sugar, pentose means 5, so that's a sugar that contains 5 carbon atoms. And the nitrogenous base, this base, obviously nitrogenous base means it contains nitrogen as a main constituent. So mononucleotides are the monomers for both DNA and RNA. How they're different, how the structure of the DNA is different from the structure of the RNA, that's a direct question that could be asked in the A-level. Very simple, very easy. Most of students get this straight ahead. So the pentose sugar is in the molecule of the DNA is called deoxyribose. Deoxyribonucleic acid. So the pentose sugar here, the sugar that has 5 carbons, is the deoxyribose. While in the RNA, it's the ribose. So that's the first difference. Check. The second difference, each DNA mononucleotide has the same sugar and phosphate group, but the base on each mononucleotide is different. What does that mean, guys? See, that's a phosphate. Phosphate is common in both DNA and RNA. The sugar is different. In the DNA, we've got the deoxyribose. In the RNA, we've got the ribose. The nitrogenous base, this nitrogenous base, guys, is made up of 4 types. 4 types of nitrogenous base: ATCG, adenine, and yes, you need definitely to know the names: adenine, thymine, cytosine, and guanine, ACTG. In the RNA, instead of the thymine, you have uracil. So uracil replaces the thymine as a base. So you've got A, U, C, and G. So that's the second difference. Here you've got the thymine as one of the bases. Here you've got the uracil. All right? So that's the second difference. Check. The third difference, it's not really directly stated here, but the third difference is DNA is made up, guys, of double helix. It's a double helix. It's made up of 2 strands. 2 strands that are twisted around each other. While the RNA, it's made up of only 1 single strand. Only 1 single strand, and that's really the third difference. So the third difference checked. That's an easy question. And you may ask, why do we need really to know the structure? Because when it comes up to study how DNA replication is happening, protein synthesis, we will really need to understand, digest, and absorb the structure very, very well. Back into the structure of the DNA. So back into the structure of the DNA. That was one mononucleotide, guys, right? So as I mentioned before, don't forget the example of the beads. These beads are connected together. Think of these, if each bead as a mononucleotide. How these mononucleotides are connected to form the DNA polymer? They're joined together through a condensation reaction. Condensation reaction between, remember, that was the phosphate group. Between the phosphate group and the pentose sugar. And definitely in the A-level, they may ask you to state exactly where the condensation reaction happens. See, it's happening between carbon number 1, 2, 3, this carbon, guys, and the phosphate group. So that's the condensation reaction. When I say condensation reaction, those who are with me in the chemistry, condensation means you remove water molecule. So you're removing water molecule in a condensation reaction just to join these two mononucleotides together. Where are these two mononucleotides? That's one, and that's the second one. Okay? Now, it goes without saying that in the structure of a DNA, for example, phosphate is the same. The pentose sugar, if I'm talking about DNA, then it's deoxyribose. But the base is different because we already mentioned there are 4 types of bases, right? So the base is not the same, it's different. You may hear about the sugar phosphate backbone, which is this. See, guys, it looks like a backbone. It's, as I said, it's the same. And then the nitrogenous base is just extended out of the backbone. So this is how it looks like. The backbone, and then the nitrogenous bases are extended out from the backbone. See, this below structure here is maybe much more clear. So, see, that's the backbone. That's the other strand. And then these are the nitrogenous bases protruding out from the backbone. All right, and what really helps for the DNA to what really helps the DNA to twist around one another and to join are the hydrogen bonds between the nitrogenous bases. So as you see here, we call this, guys, complementary base pairing. Whenever you see adenine, it's always connected to thymine. This connection's through two hydrogen bonds, okay? While cytosine is always connected to guanine through 3 hydrogen bonds. I hear some of you are asking, why is it 3 hydrogen bonds between cytosine and guanine, while there are only 2 hydrogen bonds between adenine and thymine? Because the chemical structure is different, guys. Those who are with me in chemistry, okay, hydrogen bonds forms between hydrogen and either oxygen, nitrogen, or fluoride. And the structure of adenine thymine allows it only to form 2 hydrogen bonds, while the chemical structure of cytosine and guanine allows it to form 3 hydrogen bonds. All right? So that's the case, that's the complementary base pairing. And the 2 strands are joining together through these hydrogen bonds, as I mentioned, between the complementary base pairing. And we call, this will form eventually the DNA double helix. And the 2 antiparallel, what do they mean, guys, by antiparallel? You're going to hear this term a lot. That means they're running in opposite direction. The polynucleotide strands twist to form the DNA double helix, as you see. Now, DNA was first discovered in some history part. For this history part, just guys, you just need to read it out. That's it. So in 1800, there are a lot of scientists that had timed out it that it could carry the genetic code because it has a relatively simple chemical composition. As you see here, guys, the chemical composition is very simple. Some argued that the genetic information must be carried by proteins, which are much more chemically varied. Of course, this is not a correct statement. In 1953, experiments has shown that the DNA was the carrier of the genetic code. This was the year in which the double helix structure came up to exist, which helps DNA to carry out its function. That was determined by Watson and Crick. That's the name of the two scientists who came up with the double helix structure, which is the model that was proven up till now. It's the true model of the DNA. Now, I can hear hypothetically some of you asking, where is the genetic code in all of this? Where is the genetic code? So please note, guys, that if we look into let's enlarge this a little bit. So remember, guys, we said that there are nitrogenous bases, right? So there are nitrogenous bases on extends out of the backbone. So there's a C, there's a A, maybe the next one is A, then T, then C, then G. So if you trace up one strand, the basis on this strand is what forms the genetic code. Okay, and please, you need to follow up one strand, okay? These two strands has names, guys. One of them is called sense strand. One of them is named anti-sense strand. I'll make this up clear. I'll clarify this part when we do the protein synthesis. So for now, let's say that I'm talking about the anti-sense strand. Okay, so if you trace one strand, the code may be CCGATGTT. If you have the code of one strand, you can predict the code of the other strand, which is the sense strand, right? Because it's just gonna be the opposite according to the complementary base pairing. So it's a G, G, C, T, A, C, A, A. So that's the code of the other strand, the complementary strand. Guys, that's a question that's also frequent in the exams. They may bring you the code of one strand and they ask you to predict the code of the other strand, and it's just just follow the complementary base pairing. That's a 1 mark question that everyone definitely would get. All right. Sometimes they may ask questions like this. So the bar chart shows the percentage of the bases in a DNA sample that are adenine and cytosine. On the chart, draw bars to show the percentage of thymine and guanine in the sample. So we previously know that adenine is always complementary to the thymine, cytosine is complementary to guanine. So if you know that 30% is adenine, then there must be another 30% of the DNA as thymine, because they're already complementary to one another. And if cytosine is a 20%, then guanine must be 20% as well, right? And if you add up all of this percentage, it would make up to 100, right? So that's a 30% and 30%. So that's a 60% plus 20 and 20. So that's a 40%. So it's making up to 100. I believe it's clear, guys, the connection, why adenine and thymine has the same percent because they're complementary to one another. So whenever there's an adenine, there's a corresponding thymine connected to through hydrogen bond. Same thing for cytosine and guanine. And the question number 2 is quite basic question. That's not really common on the past paper, to be honest, guys. The questions are indirect ones, but what we get the core out of the explanation. We will have time as well in the course to solve a lot and many past papers. So that's quite a direct question. Describe how mononucleotides are joined together in the DNA. I have already explained through the condensation reaction between the phosphate group and the sugar base, and describe how 2 single nucleotide strands are joined to make up the double helix. Just here, guys, again, you need to echo again the structure of the DNA and how hydrogen bonds is formed between the nitrogenous bases. Cytosine and guanine, 3 hydrogen bond. Adenine and thymine, there are 2 hydrogen bonds. Quite direct question, as I mentioned, not a very common one. So I hope the introduction for not really introduction, actually, guys, the part of the protein synthesis was this part, guys. Let me show you. This part was a critical part that we really need to make up a good base before we move into next session for transcription. So if you like the video, give it a thumb up, follow me up in my YouTube channel for more videos about transcription and translation. Thank you so much for your time. I'll see you next time.